everyone, welcome to Botany Insider. So in this lecture, we'll be dealing with one more topic from Unit 12, and that is breeding in plants. We'll be dealing with this topic in complete detail. So let's just start off the topic now. So let's just discuss about that what plant breeding is all about first. So the plant breeding is the science driven creative process. So it is a process, or we can say that it is a creative process which is actually supplied with the scientific knowledge for developing the new plant varieties that goes by the various names including the cultivar. So plant breeding is actually a technique. It is a scientific technique which is a creative process in which what we are doing is we are trying to develop new plant varieties. And this complete process of formation of the various different plant varieties could also be given the name of cultivar. Plant breeding is the science of changing the traits of the plants in order to produce the desired characteristics. Traits in simple word means features or the characteristics. So what we are doing is that in plant breeding we are actually trying to change the traits of the plant so that we could have the final plant that would be produced as a result of this creative process. The plant would be containing the desired characteristic as we want. The genes in the plant are what determines what type of the qualitative or the quantitative traits it will have. That is, as we have already discussed, that genes are nothing but the actual transcript of the proteins that would be created. So these genes would determine that what would be the quality and what would be the actual quantity and the quality of the traits that the final plant that would be produced would be containing. So the scientists and the farmers have manipulated the crops and the animals by selective breeding for the centuries. This has been used to improve the certain characteristics. For example, the most common characteristics which have been targeted upon is the food yield, that is the amount that is produced at the end of the experiment that we are targeting upon. Plant breeders work to create a specific outcomes of the plant and potentially the new plant varieties. So what happens in the plant breeding is, so this is a simple depiction of how the process continues. So initially we have the original parental phenotype, and phenotype means the physical appearance of the actual plant that we are targeting upon, and we have the parental genotype. Genotype means the genetic makeup and phenotype means the physical makeup. So in this, what we are doing is we are inoculating it with the vertically transmitted microbiota. So do not worry about this, that what it is. Just understand that we are trying to insert some kind of vertically transmitted microbiota in this particular plant that we are targeting. So as a result of the inoculation, what happens is that we have the novel F1 phenotype produced. So in the first one, we have the parental genotype plus the microbiota A, that is this one. And in the second one, we have the parental genotype and the microbiota B. So what we are actually doing is we are trying to make the final plant that we want with the desired characteristics. So let's understand about the mode of the pollination and the reproduction, and some of the examples are, so I have provided the list of self-pollinated crops, cross-pollinated crops, and some crops that are often cross-pollinated. Just go through this complete list. This might help you in some question or the other. Just pause the video here and try to note down all the points that are listed here. So now let's discuss about that how the complete plant breeding process occurs. So we have the plant that we are targeting upon. Then there occurs the genetic modification of the plant depending upon which gene we have targeted or which gene we want to actually modify or modulate according to our needs. And then after the gene modification or after the genetic modification, we have the screening for the desired modifications. That is, we would be selecting out, we would be screening out those plantlets that have the actual desired modifications carried upon. It is not possible that if we are growing 1,000 plantlets in an area, it is not confirmed that all the 1,000 would be carrying the genetically modified DNA that we have inserted. So those plants that have the desired modification would be screened out, would be taken out, and now there occurs the removal of the recombinant DNA, and finally we have the improved plant. And now this improved plant have the plant that contains a new DNA, that is the new gene that we have inserted. Second, we have the plant without the new DNA fragment but contains small modification of its own DNA, and the third one is the plant without the new fragment and also without the modification of the new plant. So the screening occurs based upon these points, that is, on one side we'll have the plants that contain the completely new DNA fragment that we have inserted. In the second lot we would be having the plant without the DNA fragments but it contains some modifications of its own DNA. And in the third one, what we have is that we have the plant without any new fragment and without any new modifications. It is the old plant itself. So we would be screening it off. We would be removing this particular plant because it does not carry the genetically modified DNA that we actually targeted upon. So let's talk about the methods of the plant breeding now. So there are basically two main methods. The first one is the traditional method and the second one is the genetically engineered method. So in the traditional method, what happens is that the traditional plant breeding process introduces a number of genes into the plant. These genes may include the genes that are responsible for the desired characteristics as well as the genes that are responsible for the unwanted characteristics. So in this, what we are doing is that we are inserting a particular number of the genes into the plant, and the genes that we are inserting, they include both those genes that contain the desired characteristics and also some unwanted characteristics. So this is how the complete process looks like. So we have the donor variety DNA fragment. So the DNA strand contains a portion of the organism's entire genome. This is the desired gene that we are targeting upon, and this is the recipient variety. So now after this complete is inserted into the recipient variety, we get a new variety with this. Now this new variety have some wanted and also some unwanted genes. So this is one of the drawback for the traditional methods. The next one is about the genetic engineering. So in this method, what happens is that the genetic engineering enables the introduction into the plant of the specific gene or the genes that are responsible for the characteristics of interest. By narrowing down the introduction of one or few identified genes, scientists can introduce the desired characteristics without also introducing the genes that are responsible for the unwanted characteristics. So in this type, what happens is we are inserting only the desired gene. So with this, we have less of labor work. Why? Because in this type of method, the screening process is reduced, or the work in the screening process is reduced. Why? Because we would not have to find out those fragments or those DNA strands in which there is unwanted or undesired characteristics also present, which is one of the drawback of the traditional method. So in the genetic engineered process, what happens is that we have this desired gene. This desired gene is taken up and this desired gene is inserted into the recipient variety of the DNA fragment, and then we have the new variety DNA strand with only the desired gene at the location where we actually wanted it to be placed. So these are the two main methods for the plant breeding. The first one is the traditional and the second one is the genetically engineered process. So now let's look at that how the various types of crops, that is the self-pollinated, the cross-pollinated and the oftenly cross-pollinated crops are actually used for the plant breeding methods. So in case of the self-pollinated crop, the first method that is used is the mass selection. So in this, what happens is that in mass selection, the seeds are collected from desirable appearing individuals in a population, and the next generation is sown from a stalk of the mixed seed. So the seeds are collected usually from a few dozens to few thousands. This process is also referred to as phenotypic selection and is based upon how each individual looks. So in this, what we are doing is we are actually selecting the seeds depending upon the phenotypic characteristic of the plant. So this process is usually used to improve the old land varieties and the varieties that have passed down from one generation of farmers over a long period to the next generation. So this is what is about the mass selection. We are doing nothing. What we are doing is that simply we are taking a lot of the seeds, and depending upon the phenotypic variety or the phenotypic characteristic of the seeds, we are selecting out some of the seeds, and these seeds are then sown into the soil into the next generation. So this is what is about the mass selection, and hence it is also given the name of the phenotypic selection. The next is about the pure line selection. So this process of pure line selection mainly involves these steps. The first step is the numerous superior appearing plants are selected from the genetically variable population. The second step is that the progenies of the individual plant selections are grown and they are evaluated by simple observations, that is the ones that are frequently occurring over a period of several years. And when the selection can no longer be made on the basis of the observation alone, the extensive trials are undertaken. In this process, there involves careful measurements to determine whether the remaining selections are superior to the yield ability or to the other aspects of the performance. So in this, what we are doing is, in this also we are using the phenotypic characteristics only, but in this what we are doing, first we are taking the numerous superior appearing plants are selected, and these superior appearing plants are then sown into the soil, and then we have the progenies of the same. And after these progenies are grown, what happens is they are evaluated by simple observations. And after the observation, if cannot be made on the basis of observation itself, what happens is we use some extensive trial for the same. So this is all about the pure line selection. The third one is about the hybridization. So the main objective of the hybridization is to combine the desirable genes that are found in two or more than two varieties and to produce the pure breeding progenies that are superior in many aspects to the parental types. So in hybridization, the phenotypic characters are not taken into the prior consideration. In this, the desirable genes are actually selected, and they are selected depending upon whether they are found in two or more than two varieties. And if they are found, so these desirable genes are used to produce the pure breeding progenies that are superior. These progenies that are produced as a result of the combination, so these progenies are superior to the parental types also in many respects. That is, these pure line progenies must be having some extra advantage or some extra characteristics as if compared to the parental types. Let's discuss about the inbreeding, that is the next process. So make sure you remember about that what is inbreeding, and then we'll be discussing about outbreeding also, because questions have been framed many times from inbreeding and outbreeding alone. So what is inbreeding? So the word itself says its meaning. Just break the term into two. So we have in and breeding. So in this, what is done is we are actually mating of the plants that are genetically very closely related. That is, what we are doing is we are trying to mate those progenies that are very closely related to each other. So as a result of this, what happens is that it leads to the increase in the homozygosity and it leads to the decrease in the heterozygosity. What is is homozygosity and heterozygosity? I'll be explaining you in a bit. So inbreeding helps to maintain the pure line and it helps to maintain the original character. And the most common example of inbreeding is the self-pollination. So now what is homozygosity and heterozygosity? We need to first understand about that what is homozygous and heterozygous. So as we study in the genetics unit, we have some genes and the alleles. So if we consider these two, so it is one gene and the two variants of it are known as the alleles. So capital A and capital A are known as the alleles of the same. So we have two varieties of it, either the dominant one or the recessive one. So if the both dominant and dominant one are together or the recessive are together, we have the condition of homozygous. Homo means same and zygous means zygote. So when we have the same alleles together, they would be called as homozygous, and when we have a combination of the two, so we would be calling it as a heterozygous. So how the inbreeding helps to increase the homozygosity? Let's try to understand it now. So as we have discussed that capital A or small a is what is called as homozygous. Capital A small a leads to the heterozygous condition. So how inbreeding helps in increasing of the homozygosity? So consider we have a plant of small a and the other plant is of capital A, and when we crossbreed it, so what would happen is that we will be having capital A, capital A, capital A, capital A, capital A small a and capital A small a as our first generation progeny, that is the F1 progeny. So as you can see, in the parental condition we had one homozygous, but in the F1 progeny we had two homozygous. So we can see that it helps to increase the homozygosity of the generation. And how it leads to the decrease in the heterozygosity? So as and when we would be mating the F1 progeny with all together, in the subsequent generations we would be noting down that there would occur decrease in the heterozygosity, that is there occurs decrease in the condition where we would be having capital A and small a as the genotypic makeup of the generation that we are forming. So this is all about the homozygosity and the heterozygosity. I have tried to make it as simple as possible. I'll make a detailed video of the crossing in the genetics in the future videos as well. So kindly wait for those videos. I'm working on them. But for this particular video, just understand the basics of it, that what is capital A, what is small a, and what are their combinations called. So now let's discuss about the disadvantages of the same. So talking about the disadvantages of the inbreeding process, what happens is that in inbreeding there occurs low yield. It could either lead to inbreeding depression, it could lead to the heterozygous condition in the subsequent generation, and there occurs less variation the inbreeding process follows. So as you can see in the figure also, we have capital A as our dominant allele and the small a as our recessive deleterious allele, that is it is somewhat dangerous in nature. In the beginning we have the parents which had capital A small a and capital A capital A. So capital A small a is about the heterozygous as we have discussed. Capital A is of the homozygous condition. So now as and when they are interbreeded, when the inbreeding occurs, we would have the final result as capital A, capital A small a, capital A small a, and one more capital A. So now as and when these two, that is capital A small a and capital A small a, are interbreeded together, we would have one combination as small a, which is very dangerous, which is deleterious in nature. So this is what is inbreeding depression. As and when the inbreeding process is carried on for many generations, there occurs depression, there occurs inbreeding depression, and as a result of inbreeding depression, what happens is the deleterious alleles tends to come together and it leads to the low yield, or it leads to the lesser amount of the individuals that are being prepared or that are being produced gets lowered down. So this was about the disadvantages of the same. So now let's understand about that what is outbreeding. Till now we have discussed about inbreeding. Let's discuss about the outbreeding now. So outbreeding, as the word suggests, it contains two words, out and breeding. So in this we are mating the unrelated individuals, and this process is also called as the crossbreeding. So in this what happens is that we are actually mating the very unrelated organisms all together. So as you can see in the figure, in this what we were doing is that we were actually mating the capital A small a with capital A capital A. So as these two are not interrelated together in any way, so this is outbreeding, and as a result of outbreeding we had capital A small a, but this is not deleterious in nature as of this condition, hence it is more safer to have outbreeding. There are mainly three types of outbreeding processes. The first one is about the intraspecific, then we have interspecific, and the third one is intergeneric. So in case of intraspecific, there occurs mating between the members of the same species. In case of the interspecific, the mating occurs between the members of different species, and in case of the intergeneric, in this the mating occurs between the members of different genera that contains very different genes altogether. Let's talk about that what are the advantages of the outbreeding process. So in this what we are doing is that there occurs increase in the heterozygosity, there occurs increase in the variation, there occurs increase in the yield of the products that we are focusing upon, and also it leads to the production of more grains. All the disadvantages that are faced in case of the inbreeding are all covered up in case of the outbreeding. So what is heterozygosity? I have just discussed in case of inbreeding. So make sure you have a grip on this. So now let's discuss about the last topic, and that is heterosis. So what is heterosis? So heterosis in simple word means increased growth vigor or the yield of the hybrid plants over the parents. So what we are doing is that in heterosis, what it is, so it actually means the increase in the growth yield or the growth vigor as of the hybrids in comparison or over the parents. It is also called as hybrid vigor. Heterosis refers to the phenomena that the progeny of the diverse varieties of a species or the crosses between the species exhibit greater biomass, the speed of development, and the fertility than both the parents. It leads to the superiority in the F1 generation, which decreases in the future generation. In this, nothing but what is happening is that there is more of yield in the F1 generation or any generation that we are focusing upon. But this increase in the yield is seen only in that particular generation, and it decreases as we go towards the future generations. And also it is a phenomena in which what we are actually doing is that there occurs increase or there exhibits greater biomass, and also the speed of development and the fertility is more than that of their parents. So now let's just discuss that where all the heterosis actually works. So it leads to the altered transcriptome and the gene expression. The phytochrome signaling and the stress response also gets altered. Then we have light and the energy use efficiency getting increased. The circadian clock regulation gets increased. There occurs increase in the sugar and the starch level of the biomass. The photosynthesis, carbon allocation, and the nitrogen allocation, all of them gets improved. And also there occurs increase in the DNA methylation and acetylation. So what is phytochrome? So these are the plant hormones. We have completed the plant hormones in detail on the channel itself, so make sure you watch those videos as well. We have also completed the biological cycle or the various types also, I'll put the links of the same in the description. And also we have completed about that what is photosynthesis and also the nitrogen cycle and its complete regulation in full detail. I'll put the links of all these videos in the description. And talking about the advantages, so it promotes early maturity, it leads to the uniform size of the fruit, it is for the resistance of the biotic and the abiotic stress, and it always produces higher yield. Talking about the disadvantages is of higher cost of production. There occurs fresh seeds in every season, and also sometimes these seeds are also vulnerable to the diseases. So with this we have completed the complete process. Talking about the application of the plant breeding, so the plant breeding is applicable in very vast areas. That is, for example, it is applicable in genetics and biotechnology, in the robotics, biotechnology, bioinformatics, genomics, statistics, agronomy, and also the horticulture, and it is also involved in the plant pathology and the entomology. So with this we are done with the complete topic of the plant breeding in plants in complete detail. So I hope this lecture was helpful for you, and if you like this video, please do like, share it with your friends, and please subscribe to my channel. And also I'll be coming up very soon with more informative videos as well. So thank you so much for watching, and I'll see you soon. Bye.